Recording begins. This is October the 13th, 2025, and we are in Biology 105. Boo! It's October. It's very scary, all right? But we're going to try to keep it together for 50 minutes to enjoy this lovely Biology 105 lecture, which I have prepared for you today. It is week 8, all right? That puts us at the halfway point of the semester. Regardless of how you do the math, we're halfway there, all right? So we did it. Great job, everybody. Now just maintain this level of energy and work ethic for another 2 months. Thank you. I appreciate you. All right. Here we are in week 8. What do you have to do? This is one of our busier weeks. There's more assignments due this week than usual because of the online lab midterm. The online lab midterm, though, it's available now. All right? It opened up this morning at 12 a.m., okay? And it only covers weeks 1 through 7 of the labs. It's open notebook, all right? It's online, so you can use whatever resources you want, right? There's nothing I can do to stop you, okay? But as I keep telling you, you want to try first to answer these questions using your lab notebook, because that'll let you know if your lab notebook's thorough enough. You know what I mean? Question. One attempt, it is worth 50 points. Yep, you get one shot on it. So, what I suggest is you have your lab notebook open and you look at the question and go, oh, my lab notebook's not thorough enough, I couldn't find this answer, and then look it up somewhere else. Yeah, instead of, going for the guess. Awesome. Yes, question. I believe that it's 2 hours. I'll double check that for you. It might even be 3. I have to go to the build page, and then I have to go here, and then I have to go to the settings, and then I have 2 hours. Yep, you get 2 hours once you start it. There's 1 attempt. Ooh, somebody's already done it. Thank you. All right. Cool. Also, oh yeah, question, please. And then I see a question. I think it's 12 questions, multiple choice, or, drop down windows. It's pretty short. It's basically like a question or 2 from each lab, Yeah. Most people who do it complete it in about half an hour, Yeah. Yes.
Is it going to be split as like a homework grade or a user grade?
All of the points in this class are weighted the same. So 50 points is 50 points regardless on if it's an exam or a reading quiz or a lab assignment. Yeah. Solid? I love it. Awesome. Yes. No, it's only week 1 through 7. So you could do it now as long as you understood the polyphenol oxidase enzyme kinetics discussion. You'll be fine to answer all the questions on there. Awesome. This is all great. Any other questions about that? So yeah, that makes this week a little busier than usual. So don't let, I would say if you can, try to get the lab midterm done earlier in the week so you don't get swamped. It's only open until Friday as well. I really want you to get it done this week. All right? Cool. Now the other assignments, photosynthesis graphing assignment. You have to graph some data that you're going to generate in the lab. I have a tutorial uploaded as per usual with the video recording of me doing the Excel work. I'm just really hoping that having you do all this Excel stuff will get you comfortable with Excel when I eventually have you all in my future classes and I make you do a ton of Excel. It saves me work later. You see, it's entirely selfish. It's not about you developing important life skills. It's about me having to work less hard. All right. I know that's true. I'm just having a goof with you all. Quiz chapter 10 is also due this week. All right. Those are the things that you have to do this week. Questions? Awesome. Very cool. We're going to try to get through respiration so we can get into photosynthesis, get all caught up. I'm pretty excited about it. Everything's linked here in the week 8 module, reading, recording, photosynthesis explained video, laboratory materials, week 8. Oh, if you want to be super prepared for the lab this week, You could watch Bridget and myself running the lab in this video, which I found from back during quarantine times when we were doing virtual labs. It's you get to see me without a mustache. It's great. It's very strange for me as well. Cool. Anything? It feels like we've been on vacation or something. I'm having a hard time getting my momentum up. Are you all ready to start do the lecture? You excited? All right, let's do it. Here we go. Thank you so much. Going to downloads, going to respiration, week 7. It's very exciting. Here it is. All right. Let's pop open this slide right here. So remember, there's an embedded video that you can watch right here if you want to check out, a video of cellular respiration with models moving around and everything. There's a ton of cellular respiration resources. It's a cool thing to understand. I'm going to explain it to a pretty basic level with a lot of focus on oxidative phosphorylation, because that's a common process between both photosynthesis and respiration that also re-emphasizes the importance of membranes in providing structure and membrane permeability concepts. It really brings everything together. So we're going to breeze past glycolysis, pyruvate oxidation, and kind of breeze past the citric acid cycle as well, okay? But there's still stuff you need to understand about them. So ask lots of questions if you'd like, and we're just going to get into the 3 stages of cellular respiration. Here we go. Glycolysis. First thing you do is you take a 6-carbon sugar glucose and you split it into 2 3-carbon molecules of pyruvate. The gray circles in this figure indicate carbon. All right? You go from 1 big molecule to 2 small molecules. The net ATP gained here is minimal. It is via substrate level phosphorylation, where there's all these steps of phosphate groups getting attached onto this molecule, and then phosphate groups getting ripped off of this molecule by enzymes. So it's all substrate level. I'm just saying that because all you really need to know is, glycolysis gets you a little tiny bit of ATP via a process called substrate level phosphorylation. That's all you need to know. But I've never felt great giving that lecture and then just saying, it's a process called substrate level phosphorylation. So if you want to know more about it, I've got more information on these slides. We're not going to talk about it very much, though, because as I said, I'm going to be focusing on oxidative phosphorylation, which yields the majority of ATP from cellular respiration. So, your takeaway. Big molecule of glucose gets split into 2 smaller molecules of pyruvate. You get a little bit of ATP, just a little bit, and you get some NADH. NADH is an electron carrier. So NADH is that electron carrier in its reduced form, by which I mean it has electrons on it. It took the electrons from glucose when those bonds got broken, all right? And now it has those electrons. And that's going to be important later. A little bit of ATP and a little bit of NADH. Cool? Awesome. Yes, please. No carbon dioxide is generated here. Very, very good. I'm going to give you 3 John points for that. Check it out. There were 6 carbons, and there are still 6 carbons. All right? So when we go to smaller molecules, the carbon gets split off. It'll go off as CO2 later during the citric acid cycle or the Krebs cycle. Sick. All righty. So substrate level phosphorylation. Here's a better pathway that has more details. It shows you all these intermediates and how one of these things exists at an equilibrium, and all this stuff moves out of this pathway, and it's way too complicated. So we're just going to breeze right past it. But usually the BIOL 105 thing we tell you is you have to put a little bit of energy into the glucose molecule. You invest a little ATP, but you get more ATP than you put in. All right? So technically, you gain 4 ATP in glycolysis, but it takes 2 ATP to kick glycolysis off. Not a thing that you need to be super concerned about, but something that you're welcome to become interested in and look into further. Okay, stage 1, done. Where did it happen? It happened in the cytosol. For my own memory, I just oversimplify this for myself and say, hey, glucose doesn't fit across the membrane, so glycolysis happens in the cytosol, and then the pyruvate can be shipped into the actual mitochondria, or mitochondrion in the singular, right? And that has always worked for me to remember that glycolysis occurs outside of the mitochondria. All right, question, please. No problem. Glucose is and once again, this isn't entirely true. Glucose is too big to cross the membrane of the mitochondria. So first it gets split into pyruvate, and then pyruvate gets shipped into the mitochondrion where it can be broken down the rest of the way. Make sense? Awesome. Very good. Okay, stage 1 is stage done. We've got ATP and NADH from it. Time to go to stage 2. Here's NAD plus and NADH. It carries electrons. It's an electron carrier. Don't try to memorize the structure. It's not essential for your understanding of this process. But remember, these things carry electrons. They're called electron carriers. The electrons are going to be needed in the final stages here. All right. Very good. Energy investment, energy payoff phase. We talked about it. Energy investment phase and energy payoff phase. We got it. We got it. We got it. We got it. There it is. Pyruvate oxidation. Question, please.
So it invests 2 ATP but forms 4.
Yep. You get 4 net ATP. Oh, sorry, 2 net ATP because you invest 2 and you form 4.
Okay, so net of 2. Okay, great.
You got it. No problem. And remember, I'll never be quizzing you on you get 2 here, you get 2 here, and you get 28 here breakdown. It's all about where does the majority come from, because those numbers are not accurate, okay? That's like the ideal maximum yield that you could possibly get, and the ideal is never real. Whoa. All right, oxidation of pyruvate. So I'm going to give you detail here beyond what you necessarily need for a healthy understanding and a healthy building of a model for this process. So here's the point of this. Stage 2, it's broken up into 2 steps. In 1, you get pyruvate oxidation. What does that mean? Well, this 3-carbon molecule is going to be oxidized. So an electron is going to be pulled off of it. Remember our redox reactions? And when that electron gets pulled off, that breaks a covalent bond, and you lose one of those precious carbons, and here you get some CO2, okay? You get a CO2 from pyruvate oxidation, okay? Pulling that electron off is going to also let you get some NADH. It's a high energy electron, pop it onto NAD+, and then take that electron and shove it into another process later. Cool? So this first step of pyruvate oxidation, it makes this molecule called acetyl-CoA. And then acetyl-CoA, which is now a 2-carbon molecule because pyruvate got oxidized, acetyl-CoA is now going to go into the citric acid cycle. All right? And what's going to happen in the citric acid cycle? It's going to break acetyl-CoA down the rest of the way. So stage 2, pyruvate oxidation and citric acid cycle, or the Krebs cycle, right? Stage 2 finishes breaking down the glucose. That's the key takeaway, is that at the end of stage 2, all of the glucose has been broken down, even though technically this is a cyclical process, and so it's like more complicated than that. I'll show you a couple of diagrams of it. My key takeaway for you is that this finishes breaking down glucose, and you've pulled all of the energy out of it now. The question is, where does that energy now reside? Some of it's been lost to the system as heat because it's been converted. But the majority of that energy is not in ATP. It's actually in those electron carriers, that NADH, and to a much lesser extent, this FADH2. So you've broken down glucose, you've pulled the energy out of it, and you've stored it in these electron carriers and a little bit of ATP. Now in stage 3, you're going to use all the energy from those electron carriers to get a ton of ATP, all right? But here at the end of stage 2, the glucose molecule has been completely broken down, and the energy is now stored in NADH, FADH2, and ATP. All right? Kind of. It's okay to have this one as a kind of. My biggest takeaway for you is that what the hell are all those electron carriers going to do? What are we going to do with all those high energy electrons? That's what I want you to be thinking about right now. All right? Because that's what's going to feed into oxidative phosphorylation, the only stage that we're really going to go in depth on. All right? Stage 1, stage 2, break it down. Stage 1, glycolysis, it happens outside of the mitochondria. Stage 2, finishes breaking down glucose, gets you NADH, ATP, a little bit of FADH2. That's it. Cool? All right, question, please.
Energy is also stored in the NADH and FADH2. Is this because they're electron carriers?
Yes, exactly. So check this out. The electrons have energy, and when you throw the electrons onto something, you give it the capacity to have more order. You're giving it more energy. So whatever gets reduced now has some energy. Now sometimes it's a very small amount of energy, like when oxygen gets reduced and becomes water. It depends on if the electron was very high energy before it reduced there, right? So that's going to be the next tricky step. Exactly. 100% accurate. It's all about the energy levels of the electron, because the electron can exist at a low energy state really close to the nucleus, or at a high energy state very far from the nucleus. And if we blast something with energy, it'll kick the electron out, and if the electron falls back, then it sort of restores itself. But if the electron goes too far out, it can just be blasted out of there. All right? So yeah, we're going to be talking about energy levels of these electrons as well. Yeah, of course, it's great stuff. So far I've really just been talking about electrons as, oh, it's like a battery, and I can put it onto something and now that thing has energy. But we're just about to hit the point where we talk about a fully charged battery versus a mostly depleted battery. Okay, here we go. This is the actual citric acid / Krebs cycle that we're going to check out. We're going to look at it right here. All right. And I just want to keep reiterating to you, not our main focus, because cyclical reactions like this, they're always like, this is super oversimplified. Yeah, it's not the purview of this class, okay, to focus a lot on this, in my opinion. So yeah, you fully oxidize the glucose. You take the electrons off of it, you break it down into carbon dioxide. That's my big takeaway here. Look, there's a bunch of steps. CO2 comes off, you get NADH, you get ATP, you get FADH2, you get NADH. It keeps going over and over and over and over and over and over again. All right, we've got this oxaloacetate molecule that you start with, and then you attach this acetyl group from the pyruvate oxidation, which is attached to this coenzyme, and that makes acetyl coenzyme A, because that's essential for the enzyme that fixes this acetyl group onto oxaloacetate to make the citrate molecule, which is now another 6-carbon sugar, and it's going to slowly break down until it breaks down into oxaloacetate, which then again is fused with the remnants of a glucose breakdown, a pyruvate oxidation, okay, and then you go again and again and again and again, and it's a cyclical process, and it's fascinating. We all loved it, right? Cool. Yeah, it's good stuff, right? Now, there's all sorts of really, really fascinating things, like, hey, this malate's actually a super important molecule. And malate sometimes leaves the system and enters the system. And oxaloacetate, is it all just, whoa, I just blasted somebody in the eyes with a laser, sorry. Is oxaloacetate and malate, are these all just sticking in the mitochondrion? No, okay? And sometimes these molecules get shipped out of the mitochondrion and then are used to rebuild glucose. It's all infuriating, okay? So we're not gonna worry about it. We're just gonna accept that biochemistry is a beautiful thing, and we all can't wait to become biochemists. It's gonna be so much fun. I hope you have a great time doing it. For me, I'm gonna stick to a figure like this, and I hope you'll all join me on this journey. So now we basically have the same figure we've been looking at with a couple little additions, all right? One addition is, hey, all the CO2 generated by respiration comes from here. Glycolysis didn't make any CO2 because you went from a 6-carbon sugar to 2 3-carbon molecules. Okay? Pyruvate oxidation citric acid cycle, stage 2 of respiration, produces the carbon dioxide. A little bit of ATP, a little bit of ATP. The vast majority, bless you, the majority of the energy that was in glucose is now here. I know it's repetitive. I know I've been saying the same thing over and over again, but it's because this is what I want you to go out into the world remembering. It's the stuff that'll really be useful when you sit down to look at these concepts in more detail. This is the foundation, all right? That's why I'm emphasizing it so much. Solid? Are we all, oh, cool, awesome, awesome. All right, we're all good with stage 2. We're ready to talk about stage 3, where we'll actually get into some details. It's the more complicated one because I'm going to explain it in more detail. I mean, these are complicated, but they seem less complicated because I tell you not to worry about the details. Anyways. Pyruvate oxidations happen, boom, 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 boom. Krebs cycle, here it goes, la, 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 la. Here's a more involved Krebs cycle. We're not worried about it. Let's keep on going. Fascinating stuff about pyruvate becoming oxaloacetate. There's all this cool stuff. Malate can leave, reverse glycolysis. You use it to build more glucose. That's what I was talking about before. It's all very interesting. You will not be tested on this. All right. It's just meant to be like, hey, look, there's more. Okay. John, you're repeating yourself too much. Everybody knows that you only care about oxidative phosphorylation. You've made it clear from the beginning. And who cares about anything besides oxidative phosphorylation in cellular respiration? Really? You like memorizing pathways and stuff? No. We want to talk more about like physics and a proton motive force, because we love the fact that the membrane is impermeable to protons. And you've been promising, I'm speaking as one of you right now, you've been promising us that the membrane's impermeability to ions was key to survival, and that you'd explain that to us. And now I'm finally gonna do it. Don't worry about the rest of your seat, because you're only gonna need the edge as we dive into oxidative phosphorylation. All right. The energy driving oxidative phosphorylation comes from these electron carriers. Here we go. What's gonna happen is those electrons are going to start at a very high energy level, and in a step-by-step process, they're gonna keep falling down to a lower and lower energy level, and every time they fall to a lower energy level, that energy is gonna be used to pump protons, okay? You're gonna be pumping protons into the intermembrane space. I got a diagram to show this. That's gonna generate a proton gradient, which then ATP synthase harvests to synthesize ATP, all right? So you got all these high energy electrons. So you've got this electron transport chain where the electron goes boop, 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 blah, and it ends up as a very low energy electron. As that electron falls down, you're throwing protons across a membrane, all right? And those protons can't cross the membrane again unless they go through ATP synthase, which sort of acts like a hydroelectric dam. It's like, and this was once proposed as a way to store energy without having to use batteries, it's like building a dam at the bottom of a giant cement chute, and then when you have solar power, you run that dam in reverse to pump water up that chute. Then when you need the power, you just let the water flow back through. All right? It's very analogous to that sort of process. Let's take a look at it now with a diagram. So, NADH, FADH2, both, well, NADH from stage 1 and stage 2 of cellular respiration, FADH2 from stage 2 of cellular respiration, specifically the citric acid cycle. All right, they're carrying electrons at a very high energy state. NADH is better because the electrons are higher energy on it, but FADH2 is nice because it lets you harvest electrons that were a little lower energy. So they're all starting at a very high energy level. The electrons go from a higher to a lower 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 to eventually their lowest energy level when they slap down here onto oxygen. Okay? And that energy that is lost, well, it's lost energy. It's not just destroyed. It's used to pump protons. Which direction do the protons get pumped? Well, they go from the mitochondrial matrix, the innermost chamber of the mitochondrion, to the intermembrane space. All right? Cool. Now, when this thing lived freely, when the mitochondrion was a free living organism, it might have just been pumping these things outside of itself. So it makes its internal, its cytosol basic, all right, and makes its surroundings a little more acidic. But this process is way more efficient if you can pump those protons into their own chamber where they can't just diffuse away. That's not important for you to, put together, but it's interesting. Anyway, intermembrane space. You pump the protons here. Okay, they come from the mitochondrial matrix. Technically, this is going to lower the pH of the intermembrane space, making it acidic, and raise the pH of the mitochondrial matrix, making it basic. Technically, that's what's going on. All right, you pump these protons, you concentrate them, then the only way the protons can cross the phospholipid bilayer, and the system wants to fall to equilibrium, right? So you force the protons to flow through the rotor, okay, that is ATP synthase. So the overall process is called oxidative phosphorylation. There's a bunch of redox reactions happening here in the electron transport chain, and the end result is you phosphorylate ADP, making ATP. Oxidative phosphorylation. All right? Good stuff. Oh yeah, one second. It's two sub-processes. The electron transport chain and chemiosmosis. So the electron transport chain plus chemiosmosis equals oxidative phosphorylation. Question, please. No problem. It's like an umbrella term.
Can you explain that stuff a little bit more? Like, so from my understanding, the electrons go down to the lowest energy states, and then because of this, it's able to come out into the intermembrane space. Like, I guess I'm a little lost.
Yeah. Oh yeah, no worries. So think about it like this, right? This electron was at a high energy state here on NADH, and now it's a lower energy state on this quinone. I forget exactly the name of this electron carrier. There's so many. Some class might eventually make you memorize them all. But when the electrons go from here to here, they went from a high to a low energy state. That's an exergonic reaction. So there's released energy, right? It has a negative delta G value. The energy released is used to do work, and that work that's being done is active transport. So you're getting energy from an electron falling from a high to a low energy state, and you're using that energy to actively transport protons across this membrane. Remember previously we talked about like how, oh, ATP is an energy currency and is usually used for work like this. Well, there are other energy sources that can also drive work like this.
Sorry.
No problem, yeah.
So the energy is released to do work, which acts after transport. But you also mentioned a chemical, a gradient.
Yes. So when you pump the protons, here, can I do... It's not very visible, is it? Document camera. Boom. All right, let me try this again. This is my very lazy phospholipid bilayer. These are supposed to be the hydrophilic heads, and these are the hydrophobic tails, right? Phospholipid bilayer. Beautiful. Flip it around. Okay, so you are letting these electrons fall to a low energy state, and that's pumping protons, right? Remember that these protons cannot diffuse across the phospholipid bilayer. So effectively, you are increasing the concentration of protons on this side. You are decreasing the concentration of protons on this side. You are setting up a concentration gradient.
So the water repeats so that also helps with the transport.
Yes, it does get confusing because it's like, well, yeah, but we already talked about diffusion, and it's all about the movement of these molecules. What makes it actually have to go through this channel, or why would it go through that channel? But I'm just gonna urge you to please not think about it too much while we're laying down the foundations. Definitely think about it again later, because this thing can also run in both directions, and ATP synthase can be used to spin that rotor to like drive movement of a flagellum. There's all sorts of cool stuff going on here, but we're at like basic level for now. Yes, a question.
So the electron transport chain powers the work that pumps protons into the intermembrane space. And then the protons move from the intermembrane space back to the matrix through the ATP synthase channel.
Yeah.
That's how most of that ATP is synthesized.
Exactly. So you set up the concentration gradient, which is a form of energy. Order is energy. It's voltage. It's all these different things. It's just, I mean, really, the best analogy, honestly, is a dam at the bottom of a chute, right? Does this look like a dam at all? I should have figured out. I should have thought this one through a little more. There we go. It's a dam at the bottom of a chute full of water, all right? You can run this dam, you can use energy to pump water up here and then harvest the energy by letting it flow back through here. And why would you ever do that? Why not just use the energy? Well, imagine that you're using like wind power, okay? Why am I drawing a turbine? This is... I'm too deep in now, I can't stop. Imagine that you're using wind power or a solar panel. That's a perfect, oh man. Sometimes, you know, forget how good I am. Run those cables, boom. Run those cables, boom. Now I promise this is gonna all be worth it, all right? So you use the energy when it's available, all right, to pump this, to pump this here, and then you can let it flow back through. Here's the problem, right? There's a ton of energy coming from the sun. You can only harvest a little bit of it, you see what I'm saying? And it's not always instantly available. So I don't want to get too caught up because that makes us think about photosynthesis. Yeah, the analogy is going to get away from me immediately. But check this out, right? This is energy in one form. You're using it to store energy in a different form. So you went from high energy electrons to a concentration gradient, and then you're harvesting that concentration gradient through ATP synthase. Does that sort of make sense? Okay, sick. I love it. A little bit? Okay, that's okay. Cool. And there's additional pieces here of like that electron is at such a high energy state to begin with that if you just tried to harvest all of the energy from glucose at once, the cell boils and it explodes. So you have to take that energy and change change forms. You have to slowly, piece by piece, let those electrons fall down to lower and lower and lower energy states. Because if they fell all at once, your cells pop, all right? So you change the energy form into this concentration gradient so then you can harvest it in a more controlled way. That's another way to think about why this process has to be so complicated. Yes?
The universe is used between the electron and the proton.
Totally.
So
Yeah.
you're pumping protons instead of electrons?
I don't know how you could possibly be confused. You're using the energy state of electrons to pump protons. No, it's super easy to get mixed up here, 100%. So what you're doing is you are letting an electron fall from high energy to low energy state. As it loses energy, that energy is being used to pump protons. Why? Because you cannot harvest all of that energy at once. If you strip all the energy off of glucose all at once, that's what happens when you like burn sugar. If you ever like lit, kind of, yeah. You have to do this process in a lot of annoying steps. You have to break down glucose slowly, all right? But then as you're breaking it down, you store that energy in a safe form of NADH and FADH2.
So proton and electron is a form of energy?
The proton, oh man, yeah, the proton concentration gradient is a form of energy because any concentration gradient is a form of energy.
Okay.
It's really super frustrating.
Molecules have both protons and electrons. You can't have them. Right?
Kind of. This is a specific type of energy here called the proton motive force. And it really works because protons can't freely cross here. But whenever you have a battery works because you have concentration gradients of charged particles. And you force them to move through a wire.
The only way those protons can be forced through is by the electrons losing energy.
The electrons using energy, yep, pumps these protons actively. This is active transport here. So you're consuming the energy that was in these electrons. Yeah, but you're doing it bit by bit because you can't release it all at once. So you set up this concentration gradient. It can handle it. It's like the biological system has to be very careful and have a lot of safeguards in place. That's one of the reasons why this is so inefficient. Yeah. Well, yeah, everything's pretty much being used. In fact, a great example of like the no waste thing is right here at the end when you're going to actually be consuming hydrogen to make water inside of the mitochondrial matrix, because that's going to further draw down the concentration of hydrogen atoms or hydrogen ions inside of the mitochondrial matrix. And the greater the concentration gradient, the more rapidly this process can happen and the more ATP you can get. Yeah, that kind of work? Cool. Here's the problem. There's a chemical way to look at this, and a physics way to look at this, and a biology way to look at this, and then innumerable models like my dam model there, my hydroelectric dam model. And they all sort of explain it, you know? It's very frustrating. Yeah, that's really one of the issues that we deal with in science is all these fields can make accurate predictions, so their model is acceptable. But all their models don't always agree. So it's just like, okay, great. So you just pick a field, you pick a model, and you specialize, and then you make fun of everybody else's model and say that their fields are worse than yours. And then, you know, that's what you do. You become more and more insulated, and you get a lab, you teach them the same things, and then you just don't even talk to the chemists. Oh, cool. Talk to the chemists, they're good people. All right, but the physicists, no way. Okay, solid. So you can see why I set this aside as this is the thing we're going to focus on in cellular respiration, because it is very complicated. It's got a lot of different principles, and basically the same thing is going to happen in photosynthesis. There's an electron transport chain where you pump protons, and there's an ATP synthase enzyme that's going to make ATP by harvesting that proton gradient. The ATP just has a slightly different fate in photosynthesis. In cellular respiration, this is the end goal product, is you want ATP. But in photosynthesis, the end product is glucose. But you're gonna basically do this and then use the ATP to make glucose. It's really strange, all right? But don't worry, it's all gonna make perfect sense. Let's look here at the electron transport chain. So, electrons fall from a high to a lower energy state. Just slow me down with any questions, because I'm just gonna mosey through this. The electron transport chain has a ton of different components to it. There's all these protein complexes, these transport proteins that have additional helpers in them. They're not enzymes, so then these aren't cofactors or coenzymes, but they're basically all involved in holding electrons, all right? And letting electrons fall bit by bit to a lower energy state. So check this out. The electron starts at a high energy state up here, an NADH. This is the least electronegative molecule. We've talked about electronegativity already, all right? Electronegativity is how bad something wants electrons. So in order for the electron to move from NADH to electron transport, or to proton pump complex 1, okay? This molecule here must have a higher electronegativity than NADH. How does that make? Does that make sense? If this table is going to take my electron, it needs to want it more. It must have a higher electronegativity. Okay? Cool? And then if it's going to go from here to over here on this table, then this must have a higher electronegativity than this table here. Fair enough? Cool? Cool. All right. So basically what happens is the electron keeps going from the least electronegative molecule, doom, 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 eventually ending up on the most electronegative molecule, and it's going to be oxygen, okay? We've talked about oxygen before in the water molecule. Remember how polar water is? And I talked to you about how, well, oxygen's incredibly electronegative. So that characteristic of oxygen is going to allow it to play this key role in the electron transport chain of taking the electrons that are now low energy out of the electron transport chain. All right? Because check this out. You have to pull the electrons out of here, because if you don't, this whole electron transport chain backs up. If all of these molecules are in their reduced form, the electrons have nowhere to go. If these electrons have nowhere to go, then you can't generate the proton motive force. If you can't generate the proton motive force, you can't get the ATP. This is why you need oxygen. This is why if you don't breathe, you don't thrive, all right? Because oxygen is acting here as what we call the terminal electron acceptor, and the second that you run out of it, very rapidly electron transport chains back up and you stop being able to generate ATP. You got to constantly be making ATP. So here you can see that illustrated in the overall large figure. This is one of these figures, this is like a money figure right here, where it does technically have everything I want you to know about oxidative phosphorylation on it. Everything, all right? Like, hey, where does the water come from? It comes from when oxygen gets reduced at the end of the terminal electron acceptor. The only thing here is that you need to understand what any of it represents. So just looking at the figure, isn't quite enough. But if you wanted a great figure to like print out and annotate for your study purposes, this would be a really good one. How are we doing on time? We're doing pretty well. Let's see here. The electron transport chain. That was meant to sort of wrap up my electron transport chain talk. Do we have questions about that? I see thumbs. I see thumbs pointing in the good direction. All right, solid. Let's wrap with chemiosmosis, okay? So chemiosmosis, remember, you've used all the energy up now just to make this proton gradient, okay? Now, in chemiosmosis, you're going to let the protons flow back through ATP synthase. Now, the difference in concentrations of protons, high concentration, low concentration, that's called the proton motive force. It comes from a proton gradient, and it's going to motive, it's going to move something. It's going to spin ATP synthase, which just makes the enzyme change shape a bunch. And it goes click, 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 click. And then it takes ADP plus inorganic phosphate and goes click, 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 as it spins, and it makes ATP. All right, you'll learn about it in biochem if you continue. And that will be where we end today's lecture, with the chorus of walking students accompanying it. Here we go. Hey, if this thing gets permeable to protons, you die. Remember I tell you about that? There you go. Sick. Fermentation is where we're going to come back on Wednesday, and then we'll get into photosynthesis. Thank you all for your attention. I hope you have a great rest of your Monday. I'll see you. I have office hours from 12 to 1 today. I hope you all have a great day. Goodbye, goodbye, goodbye.
Why are proteins bad for the body?
Why are
Or not proteins.